Hello, and welcome to my lightning talk about my project. This year I participated in the URSA Engage program with my mentor, Dr. Sushma Naithani, and I worked on the biocuration of the rice gene network involved in abiotic drought response. So the first question you might be asking is, what is biocuration? To put it simply, biocuration is using publicly available information from sources such as databases and scientific literature to understand genes and proteins better. As researchers, we synthesize that knowledge from these sources to create a useful representation of the data. By integrating data from these sources into a database, we make it more accessible for other scientists. As you can see in the database diagram to the right, there's a lot of information to curate. This is a pathway diagram of the species I worked on, Oryza sativa. This example is the ideal of biocuration, an information-dense but relatively simple map of the gene interactions in a pathway. The research process of biocuration involves extracting and interpreting data from scientific publications. In my case, I made use of some information from Dr. Naithani's lab that they already had available and added from my own reading of scientific papers. This initial step helps us understand gene relationships and is where most of the lab work comes in. The next step in biocuration is to add in all the other information we find from other databases. One large part of this is ensuring cross-compatibility between databases. Information isn't very useful if people can't agree on what gene we're talking about. Since every database has a different naming scheme, a well-curated gene will have references to all of these names. In addition, many other factors about a gene go into a database entry, such as pathway information, location in a cell, the interactions of the gene, and the actual amino acid sequence of the protein it produces. So once all this information is assembled, where does it go? Once we have an understanding of the gene and the pathways it's a part of, this information goes into the Plant Reactome database. Here's an example of the types of information we collect for each gene before it is entered in the database. I filled out an Excel sheet like this for each of the genes I curated. I'm still in the process of integrating this information into an entry for Gramene.org. These are some of the genes I'm working with and filling out a database on. For each of these genes, I found the subcellular location, ID in various databases, and in some cases, a brief summary of the gene, among other information. This first slide shows a small summary of the upregulated genes by OSNF-YI7, where the ones inside the smaller circle are present in the nucleus and the ones outside are present in other organelles. This slide shows the downregulated genes for OSNF-YI7. Over the next few weeks, I'll be working with Dr. Naithani to integrate this information into the Plant Reactome database at Gramene.org. Ultimately, the goal is to put all this information I've collected into a format that is accessible and easier to understand for other scientists. In the future, all this information will go into the Pathway browser at Gramene.org. Finally, I'd like to give a huge thanks to Oregon State University and the URSA Engage program for their sponsorship, as well as to Dr. Naithani for her mentorship. Thank you.